Hi, welcome to Art of Academia, a weekly podcast featuring interviews with leading scientists and insider takes on life in academia, hosted by Komal and Madan, researchers from Cancer Science Institute, Singapore.
What's up? How was your day?
Day was good. Today being a Saturday.
Yes.
I think I was telling you before.
Yeah, dry lab day.
What?
It's a dry lab day.
Yeah, it's a dry lab day. It has a few things going for it. So I feel like Saturdays lately have become my favorite days of the week. So one is because I've designated this day to do dry lab stuff, bioinformatics stuff, whereas rest of the week is experiments.
Labor.
Not always experiments, but meetings and stuff. So I guess that already makes it nice. But there's more things to it. There's also like gym in the morning.
Yeah.
That adds a bit of, you have that post-workout, what do you call that? Pheromones? Not pheromones. There's some...
Endorphins.
Pheromones
is something else, Brian.
Yeah. So you have these endorphins or whatever that are secreted post-workout. So I do feel like they give you a good buzz. Normally...
Yes.
Normally I do that in the night. Because then I do and I go sleep, right? So I guess I don't...
Those pheromones don't come out.
The endorphins don't come out, yes. So I'm not awake to enjoy them.
Yes.
But yeah, apart from that also, a few other reasons. I just like my Saturday routine. So it's been quite nice.
Did you get your sunny Korean?
I did.
Very nice. Very ritual-centric kind of day.
Yeah, I go to my Korean bro and he's like, hello friend, welcome again.
Very sweet. I went, normally I join you for these S-tier Saturdays as well, but today I had a hike to go to, and I feel like this is the beginning of my hike phase for this year. For the two listeners.
It's you and me.
Yeah, us. In the last real trek that I went to, I injured my ankle, and that took a long time to recover, but now I'm back, hopefully. So we are planning to go for quite a challenging trek in Indonesia, in Lombok, called Mount Rinjani. So we are kind of prepping for that. So today we just did one trail in Singapore. And after this is going to be scaling that up, and we are going to another trek in Malaysia, in JB, which will happen sometime in July. I have texted my trek bro in Malaysia. I don't know, some of the ones which we were planning for the longest time but just couldn't figure it out. So mostly either Mount Kulai or Mount Lambak. I know them all. So that will happen. So a little escalation from what we just did today, and then hopefully this will train us enough for the real deal. We're going to also camp and stuff there in Rinjani, so quite excited for that. Some people are a little skeptical about it.
As in? Scared?
Staying on top of a mountain. But let's see. Quite excited about that.
And it's considered intermediate.
I think the last summit is difficult, but rest of it is fine. But someone did fall and die from that.
Damn, So wait, does it require equipment, climbing equipment?
I don't think so.
Okay.
Yeah, but I guess it's just the incline is a little steep, and accidents happen, of course. You're quite high, and if you slip, because it's also a little ashy. It is a volcano after all, so it does erupt sometimes, but just ashes, and that covers the ground. So now instead of just gravel, it's also ashy gravel, so it becomes more slippery. So gonna be fun.
Okay, sounds fun.
Apart from that, what else is happening? I joined a ukulele playing group.
Oh yeah, how's that going?
Very nice. I mean, what am I saying joined? I just went 2 times, but basically it's a bunch of people who meet every Wednesday at some pub, and they just jam. They just play ukulele and sing together for a few hours, get some drinks, dinner and stuff there. Very wholesome vibes. And they have been doing this for 8 years in Singapore. And the best thing is, firstly, there's no commitment. You just go show up. No one's asking you. There's no payment or anything. You just go join them. Very friendly people. And I have already learned more than what I knew. So I only knew 4 chords. Okay. That was the basis of my ukulele playing. But now I think I know 4 more.
Wait, there's so many chords?
I don't know how many there are, but
okay.
So normally what they do is they show the chords on the screen, and then there's the song, and they will write which chord where. And within the lyrics, the chords are there. So now because I am constantly doing this for 3-4 hours straight, I am practicing more. And this is something I might not do by myself. Or I have higher threshold to learn newer chords 'Cause finger placement and maybe I just don't have that skill is what I used to think. But now I feel like it's literally just practice and giving more time. So that has been great. I mean, it's making me learn and it's just fun. People are literally singing and having a good time.
So how many people show up normally for this?
10 to 15.
Wow, that's a big group.
Yeah, I'll show you a picture later. Very chill bunch of people. I was also doing paint by numbers for a good month, I think. So just basically coloring for adults.
yeah.
I was missing some art and then missing some just doing painting or something, and this was like the easiest thing to push the barrier. And it became like a great routine for me. Every night I would go paint for like good half an hour, 1 hour, 2 hours even sometimes, with some podcast playing in the back. Oh, the podcast also I was listening to was called Smartless.
Oh, I know.
Have you heard of them?
Yeah.
It's crazy, like stupid people and very funny. I absolutely love their bizarre conversations, and that was what was happening.
What's his name?
There's Bateman, Will Arnett.
Will Arnett, yes. So the
Arrested Development guy.
Yeah, there's Arrested Development guys. And I guess they also have good... I mean, so first of all, I think Will Arnett is a great voice actor.
Yeah. What gravitas in his voice, man. Oh my god.
And I guess they're also... Of course, they are very funny.
They're very good friends as well. All of them are like good friends.
Yeah, who's the third guy?
Oh, I forget his name now. Sean Hayes. I don't know who he is even. I know the two men from Arrested Development. The show I also absolutely loved at some point. They're mostly bullying Sean, like he's there for that.
Oh, is it?
Yeah. Arnett obviously is a big bully.
I listened to some of their episodes. I think I cannot remember now, but I feel like they also get interesting guests.
They do. At one point there was Matthew McConaughey, and I was gonna actually share that episode with you, but I forgot because that guy also is an amazing speaker and like just has very nice insights. Mostly pop culture people, but then sometimes some writers, some politician even, and normally they would have some context, some connection. So like all sorts of people, and it's so funny. It's just so engaging. I absolutely love it. Like the best non-brainer podcast there is. I absolutely love it. So, yeah, that was the routine for a while. What else? Anything else up with you?
Not much. We can get started.
Sure.
So this week, I basically wanted to discuss 2 papers. Well, really just one paper that came out in 2023. It's a Nature Genetics paper, and it's called Thymidine Nucleotide Metabolism Controls Human Telomere Length. So how we found out about this is Dennis, my PI, he went for a conference, I forget which one, and then he came back as he normally does with a ton of ideas that every PI typically does, I guess.
Yes.
But he was particularly excited for this one. He spent quite a bit of time raving about it. And also when he explained it to us, we were also like, oh, that seems pretty cool. And then he actually ended up discussing it for Journal Club. So that was a nice excuse for everybody to go a bit more in depth. Which is why I thought, okay, we'll take this opportunity to share with people. So basically, before we talk about what the paper actually is, let's do some background.
Yeah.
We start with telomeres. So these are basically nuclear protein structures that are at the end. Can you hear? Look, okay, let's start over. So telomeres are the caps of our chromosomes.
Yes.
So they normally get compared to aglets. Do you know what an aglet is?
No.
So it's the end of the shoelace. You know, the plastic tip at the end of a shoelace.
Wow, I didn't know it has a name, but it completely makes sense.
Yeah.
What a great analogy.
Yeah, so if you're a Phineas and Ferb viewer, you would have known it because it's a song after it. Anyway.
I see.
So why the analogy? Because chromosomes are a bit like the shoelaces.
Yes.
Where without these aglets, they start fraying apart and then falling apart eventually, right? So that's sort of what the telomere function is as well. It protects the DNA that's in the rest of the chromosome by acting as a protective cap.
Yes.
We can go into a bit more mechanism because I think that's useful.
Yeah.
The reason our chromosomes are in danger without telomeres is something called the end replication problem. So because of the way replication works, the replication machinery that copies our DNA, so I guess take one more step back.
Yeah.
Each time our cells divide, they have to copy our entire DNA and pass it on to the new cell that's being made so that it also has the information, and this can keep going.
Yeah.
And this process of copying is called replication. So like I was saying, because of how this replication mechanism works, it's a bit of a quirk that a few small bits of information towards the very end of the chromosome is lost. In just one cycle, it might not matter a lot, but if this happens over and over again, you'd keep losing until essentially all of your genetic information is gone away. So you don't want to do that, right? That's where the telomeres come in. So instead of losing this precious genetic information in the middle, you'd have this extra bits of DNA that get lost instead. So this way you can like still preserve it. But there also you get a bit of a catch. The telomeres themselves will shorten, right? So each time the cell divides, the telomeres themselves shorten, and eventually they become short enough where it's not sort of safe for the cell to divide anymore, and then it stops there. So this is typically called the Hayflick limit. So basically it's the number of times a cell can divide until it can no longer divide. So this controls a lot of things. For example, aging. This is one big reason why we kind of don't live forever. It's not the only reason, but it's one big factor in this, the number of times we can divide, because of telomere length. And then, okay, so now the final piece of the puzzle. Some of our cells, let's say like stem cells or even our germ cells, so these are specialized cells, they kind of have to keep dividing. These are highly proliferative cells, and our health sort of depends on them being able to divide a lot, much more than what these typical Hayflick limit allows them to. And how they do that is with an enzyme called telomerase. So this is a protein which can basically synthesize new telomeric DNA towards the end of our telomere. So basically, if the telomere shorten, telomerase come and it can re-lengthen it, allowing these cells to divide more. Okay, so those are all the main elements in normal healthy cells. Okay, so let's go into the diseased setting. So now there are a class of diseases broadly called telomere biology disorders. And the reason it's so vague is because we actually don't know the exact mechanism for what goes wrong, except that these are sort of rare genetic disorders where the patients, they can have a wide range of symptoms, but one of the common characteristics is that the telomeres tend to be very short, and often they die very early. They cannot live very long as a result of this not being able to
replenish these
telomeres or these stem cells that we mentioned earlier. So, okay, so then now this is one good reason as to why it becomes important to understand how telomere elongation works, right? So then if you know that, you can try and kind of cure these patients.
Diseases, which are dependent on the telomere length shortening.
And you know that in these patients, for example, the genes that are involved in this pathway, many of them are mutated. But still, there's like a big substantial number of telomere biology disorder patients where none of the genes that we already know to be involved in telomere synthesis, they are not affected. So that means that there's something else, but we don't know how that relates to the phenotype of short telomeres.
Okay, that's interesting actually. So how frequent or how common are these kind of diseases?
They are rare genetic disorders, but I think collectively it still affects a substantial number of patients, and it's also, I guess, made worse by the fact that it's a fairly serious disease. Like most people die either as children or very young age. Like so.
Can you give an example of a disease that belongs to this category?
I think one of them is called dyskeratosis congenita. There's many of them, but okay, I don't want to get cancelled for this, but usually the people who have it, they look weird. Normal growth, white patches on the skin.
I see. Because I have basically always heard about telomeres and telomerases in the context of cancer. But obviously because that's also because I work in this field. But it makes sense that it would also affect so many other biological phenotypes.
And it's worth mentioning there. So usually in the setting of cancer, you have the opposite problem. Cells find ways to elongate their telomeres.
So they can keep on dividing?
That's what we're talking about. But in these telomere biology disorders, the cells are not able to elongate. And that's what causes the problem.
Right.
Anyway, so that's where our paper is situated, right? That's sort of the background for it. So what the authors do here is they do a CRISPR screen. And what CRISPR screen is, so CRISPR, people might have heard about it. It's a technique where you can edit a certain gene. In this case, edit just means to disrupt its function, to make it stop working.
Yes.
And a CRISPR screen means instead of editing just one gene at a time, you have a population of cells where in each cell, one random gene is edited, is mutated, or rather...
disrupted.
Isn't there a simple word for this? Broken? I don't know. Anyway, right? Its function is removed.
Yeah.
So now you have
loss of function.
Yeah. So you have a pool of cells where each cell, is missing one random gene.
Yeah.
And now it's interesting because you can then study what happens. So for each cell, you can study what is affected because of this loss of this gene. And what the effect that you're looking for in this case is telomere length.
Right.
And they can measure telomere length by a technique called flowfish. So it's basically, you have these probes that are fluorescently labeled that can go and bind to the telomere DNA. And if the telomeres are longer, you will get more fluorescence from that cell. If they're shorter, you will get less fluorescence.
Yes.
So this way you can filter out the cells that have long telomeres. You can filter out the cells that have very short telomeres. And then you go back to the cell's DNA and you look at which gene was mutated.
Affected.
So that way you can figure out what gene is responsible for the shortening or lengthening.
Yeah.
So they did that, and when they do that screen, interestingly, they find a lot of genes involved in nucleotide metabolism.
What is that?
Yeah, so nucleotide metabolism. So nucleotides are basically the building blocks of DNA. There's 4 of them, 4 molecules, ATGC, that makes up the entire of our DNA. And metabolism just refers to a set of genes or proteins that are involved in maintaining their balance.
Yeah.
So these
they
things
either make them or break them.
Yeah, exactly. So and it's a actually fairly complicated pathway, which I also realized only when I read these papers. There's lots and lots of proteins that are involved, converting one form into another, and the abundance of one affects the other, so on and so forth. So they found that, right? And the thing is that by itself is not super novel. There was also another GWAS study, which showed that actually some of these nucleotide metabolism genes are associated with telomere length.
Right.
But this was the first time somebody had done a CRISPR screen, which is, I guess, more direct than just doing some association.
Correct.
So that part is interesting, but where the study really takes off is that once they were able to zoom in on these like nucleotide metabolism genes, they had this idea that, perhaps simply just supplementing cells in culture with nucleotides, so just putting more A's, more T's, G's or C's in the culture, does that affect telomere length? It's actually, if you ask me, is such a silly idea that I would not even have thought to try it.
Correct, yeah. I mean, when you were just describing it, I was thinking back in the paper for what was their rationale for doing this experiment. Like, I understand, yes, they see mutations enriched in proteins that affect their metabolism, but just directly putting in ATGCs is kind of wild.
Yeah, and I don't know, maybe they already knew that, right? And maybe the CRISPR screen was a way to put more context to it. I have no clue.
Sure.
But that's the way it's presented, at least, right? So they get this idea, and that's exactly what they do. They take some cells, they culture them by just adding some nucleotides and different kinds of nucleotides and different combinations.
Yeah.
And then they measure their telomere lengths. And crazily enough, so a lot of them have different effects, but the main thing is thymidine, the T, which stands out. So if you add T and T alone, along with your regular cell culture, the cells have remarkably longer telomeres. And it's not even that long. This is like after 9 days of treatment, you can already see an increase in telomere lengths, which is pretty crazy. And they show that in a few different cell lines, in a few different settings. We won't go through all of that. And the other nice thing about them, about these 2 papers, I suppose, is that they show this finding in a variety of different ways. Like they also figure out which genes or which proteins are involved when you do the supplementation, what genes are involved in actually allowing that to affect telomere length. Yeah. Right? So I would say overall it's fairly convincing story. It's not one of those poorly controlled cases. And it's not perfect. Sometimes they do miss, they are lacking replicates and all. But simply by the sheer number of different directions in which they
Yeah,
show their
the same
phenotype. It's actually fairly convincing. What else? Oh yeah, I guess then we should also go on to say, so initially they do that with just normal cells that we work with in the lab, right? So then to tie this back to the actual patients, they take iPSCs, induced pluripotent stem cells, from patients who have some of these telomere biology disorders. So this way it's representative of their biology, but you can still culture them in the lab setting. And then they supplement with DT. And turns out, like through a variety of so many different patients having different kinds of mutations in different components of the pathway, whatever, many of them are benefited by just simply adding more DT in the culture, right? And the telomeres can elongate now, which again is like a crazy result. Yeah. And I should also say that the concentration in which they supplement the DT, it's not super high, which is crucial because actually
does it match the drugs that are available which do this?
Yes, so that's one, that some of these drugs are already in clinical trials for treating other disorders, so that make them easy targets. So once they get approved, you can also try this. Yeah.
Do you think they were able to do this because of the technological advancement in any sense? Because what I'm thinking is that it's such an elegant paper, very simple, and it has only 2 authors on it.
Yeah, that was going to be one of my points. So there's 2 authors. The second is the PI. So technically it's one guy.
One PhD student.
Who did everything.
I think whenever this paper came out, they were a PhD student. I don't know what he's doing now, but it was mind-boggling to me. Yeah, but then the question was, looking at the experiments, it's literally, say, 2 or 3 techniques that they keep on
technique,
doing.
Bro.
No, like the initial, the screen
is
and
there.
then this, but then eventually it's literally that southern blot.
It's
that they keep doing.
Yes.
And add something, southern blot.
So,
remove something, southern blot.
That's literally what I wrote down. I wrote single author and one technique to rule them all.
Insane.
Yeah.
So that's what I was thinking. Like, what is it that makes it, firstly, so cool and so elegant? Is it the technological advancement, or it's literally just their thinking at this point of nobody has tried this and I don't know, for whatever reason they thought about?
Yeah, it is the latter because the technique is ancient. People have doing southern blots.
No, what about the fish flow CRISPR screen, which enabled them to see all sorts of genes that could be part of this?
So I don't know if other people have done a CRISPR screen with a flow fish, but for example, I think this sort of checking which genes affect telomere length has been done in yeast. So I think nobody had done it in the human system, sure. But still, I would say yes, the CRISPR screen is, for example, cool, but the real finding is actually downstream of that, right? Like perhaps maybe seeing the CRISPR screen gave them the confidence to pursue this in full whatever. But that's not actually the exciting part about this, right? It's what comes later, which is just culturing cells and then doing southern blot. Yeah. Which is insane. So I guess it's the latter of them just having the thought to try this, and then I guess seeing that it works, I guess also following up so much on it.
Yeah, I guess like overall for me, yes, reading this paper was super interesting given the few findings that you just summarized. But apart from that, it was also like just doing simple experiments. Like I want to now, I'm actually motivated to now go back to my analysis, whatever I have done, and then pick most interesting things and design the simplest experiments which can say yes or no to whatever hypothesis I have. Because I think this also goes back to what Ashok used to say about, and also in another podcast we have discussed of, you try to always disprove your hypothesis. So you don't need to do 10 experiments, you need to do that one experiment that will disprove your hypothesis. And yeah, I think I'm kind of motivated to just try that for a few things. Does this paper overall help you in your research, or this is just a side fun thing that you read?
Yeah, it doesn't affect the work I do in any way directly, and I really don't want to do the southern blot. It's a rather tedious technique which I don't like. And actually, if any other paper did this, right, I would have flamed them because it's just the same blot over and over again. But yeah, because the finding is so cool, they can get away with it. But I think otherwise it would be such a
absolute pain to see and do the same thing. Yeah. They wrote it also in a nice way.
Yes, it's written.
The writing
that
was also
helps a
very
lot.
simple, because I don't have background in this. I mean, of course, I'm a biologist, but there were so many new things to figure out here about the mechanism and just generally telomere biology, but very simple to understand, very nicely written.
Yeah, actually, wait, before, I mean, we've zoomed out already, but I do want to zoom in for a bit more, go back to the actual biology,
Yeah.
and just make two more points, or rather one main point, right?
Uh-huh.
So, this sort of observation that you can just put more thymidine, and then the telomeres can elongate again. So, crazy finding, but the question is how, right? How does this happen? And the sort of easiest guess would be, maybe there's just more thymidine to be incorporated. So, okay, some context. The telomeres are TTAGGG repeats. So, they need the T,
Yeah.
right? So, already that raises a bit more question of why is it that thymidine specifically impacts it so much, whereas, the A and G does not. And in fact, in the follow-up paper, you'll see that it's not just about putting any metabolites. In fact, if you put specific nucleotides or nucleosides, I should say, it actually reduces the telomerase activity instead of increasing it. So this
Like
seems to
any of the A, C, and G.
yeah, and also like the different forms,
Sure.
phosphorylated or along with the ribose sugar, whatever. So all the different variations. They don't all have the same effect, and importantly, some have a negative effect, right? So that's going already to the clue one that it's not simply about abundance.
Yeah.
But in the first paper, they do actually this very nice experiment. The broad idea is that they engineered a version of telomerase. So they added the telomerase into one that no longer incorporates thymidine. So it puts some other repeat without the T. So the T is not needed for the new edited telomerase to work. And even in this setting, when they add DT, it still does better.
That's insane.
Yeah, so this actually suggests that it's not a matter of simply having more substrate.
I was just thinking maybe it's just the first two nucleotides, and for starting off that replication or starting off addition of these primers, that's why T is needed. But now you're saying there's no T and still DT is increasing.
Yes, so it's not a matter of just simply substrate availability.
That is
It's more
insane. Do
complicated.
they have any speculations as of now? I mean, it's a very hard thing to do right now, I guess.
In the discussion they do, but to be fair, I did not look at their discussion super detailed. But importantly, they don't have any evidence yet, right? So
Sure.
that
I mean,
could
I'm
be-
sure they're working towards it, but it's very mind-boggling to
Yes.
think about.
Yeah, I can't remember if this is from the paper discussion or just from the lab discussion, but the thing was like there's some sort of competition with other. So telomerase needs nucleotides, but also the DNA polymerases need nucleotides to put, to replicate the DNA. So somebody was saying that there might be a more complicated regulatory mechanism based on the nucleotide availability. But all of that is, I guess, conjecture, and I guess they're trying to figure that out. The second paper,
Very interesting.
which was supposed to be, I guess, a bit more mechanistic, or at least that's how I thought it was. But so there they developed an assay where they can see so normally for this telomere length effects to show up, you have to culture them for a few days, 2 weeks or something, right? But they made a variation of the assay where you can really see that rather quickly. And that allows them to go a bit more in fine grain into the way the telomerase activity is affected. But even there, right, they were able to show that, you can change all these different nucleotides, they're like metabolites or the enzymes that are responsible. And all of that shows sort of what they expect. And they can go very much detailed into that. But none of it gets to the mechanistic part of what is actually causing this change in telomerase activity. So,
I mean, that is, of course, the hardest question. Like, the mechanism for any kind of phenotype is the hardest thing to address.
yeah, but even without that being said, right, like a lot of the
What's
times
what's the takeaway from the second paper?
the second paper zooms in more on this, on the guanosine supplementation stuff.
Ah, okay.
Yeah, and again, it's more about how if you put too much guanosine, it can actually inhibit the telomerase activity that you're seeing instead of supporting it, and they explain why again in a fair bit of detail.
Yeah, I guess just some more pieces of the puzzle from a different angle.
I can't remember again if this was a reviewer comment or somebody in the lab said it, but the second paper does feel like they had a bunch of extra plots remaining from the first paper and they wrapped it up into a manuscript. It's still a cool one, but yeah.
Very nice. I was also thinking about, like, after reading this paper and just generally getting the essence of it, what other papers you think of which kind of match this simplicity and conclusions that we are getting out of it in the modern day?
I see. Yeah, I can't actually think
Quite hard, right? I was also trying to think about this because it's, again, very simple. It's the same thing I'm repeatedly saying, but literally just one big experiment, and then from there the same technique. And I guess, like, these kind of very simple conclusive experiments were more in the past, like, say, discovering the DNA structure or the mechanism of replication of how it's semi-conservative, stuff like that. All these experiments we used to read in our textbooks.
Yeah, I mean, it's nice that I guess they were able to design all their experiments in a way that, it takes advantage of their expertise, which seems to be just doing TRFs, right? Southern blots. And also, I guess it is in its own way fairly creative, right? That you ask all your questions in a way that it can be answered by a single experiment. It's pretty cool.
Yeah.
But I'm not entirely sure if this should be encouraged, Maybe it's also like a quirk of this project. Like the fact that they are probably the first one. I don't know if they are the first one at least, but it seems like the discovery itself is so novel that they can get away with just doing that,
No, I mean, the thing is, the phenotype that they are testing from anything they are asking is the exact same.
Yes.
So they don't really need any other experiments at this point. It's literally length of telomere that they are asking for. So perturb whatever anywhere in the entire genome and then just see, did it change?
Yeah. Fair.
They're always constantly asking for yes-no question, which normally is not the case.
True. And maybe this goes back to, I think I mentioned this on the pod as well, right? Where in some ways, they are going deeper, but in many ways, the depth is actually sort of parallel.
Yeah.
Right? So I think the thing I was referring to is like building houses of stone rather than mansions of straw. So the idea there is that you make like small but very concrete findings first before trying to advance the story too much. And in some ways it feels like that to me because they show basically the same finding in very different,
in all sorts of ways
different
possible.
ways. Although the technique is the same, I guess they go around in the pathway. So it does make that very convincing. But true, after finishing reading those two papers, it still feels like I've read one big result and not like some, a whole story or something.
Yeah.
I guess we'll see what they cook up next. Yeah, and it's also worth mentioning that the first paper, it's a Nature Genetics paper, which is pretty big, probably as big as it gets. With the exception of, what's that, NEJM or something.
Yeah.
Okay, any concluding thoughts?
I think...
I guess we did them already.
Yeah, we kind of moved around them. Yeah. Maybe I will just quickly talk about the most immediate motivation I got from this work. It's again a different paper that it led me to. But basically the vibe is doing a single experiment which can kind of explain things to me in a simple manner. And for that, in the context of what I've been looking at, I told you last time how I'm trying to understand if you have mutation in EGFR gene, it increases chromosomal instability. But that's again very descriptive. It's correlative as well. You see a bunch of features in tumors, but you don't really know, does it give any kind of benefits to these cells, or it's just a byproduct of problematic cell segregation. So I was also reading and I found out this another Nature Genetics paper from 2024, I think, which was about looking at chromosome evolution through screens again, to see how tumor aneuploidy patterns help shape tumor evolution. So normally, how can a cell form tumor is they can either have mutations, smaller in mutations such as SNVs, so affecting one nucleotide, a few nucleotides together, causing inserts and deletions, etc. Or they can also have bigger chunks of chromosomal arms or entire chromosomes being amplified, deleted, translocated, moved around, and a lot of mess happening. But the thing is, for point mutations, a lot is known about how they could drive cancer. So genes like TP53, EGFR, BRCA, we know that yes, this specific mutation will now lead to say cell cycle changes, apoptosis, what have you. But for chromosomal amplifications and deletions, this is not really known. And I was surprised to know this is a 2024 paper, and actually nobody has kind of specifically looked at what is the survival benefit of having a particular chromosomal arm amplification or deletion. Normally, when a cell is dividing, it has some mechanisms in place to make sure that the cell is dividing properly. But if you inhibit some of these checkpoints, now the cell will divide in an abnormal manner. And that is how you get aneuploidy. So basically, they just added an agent which will add massive amount of aneuploidy to these cells. And then they let them grow for a day or two, I think 2-3 passages. And by that time, only the chromosomal gains and losses that will actually provide a benefit will survive. So there's just going to be survival of the fittest. And then eventually they take these clones, do single cell sorting, grow them further, and then eventually see how this evolution is continuing through a few months or so to see what sort of newer complications are helping these cells evolve in time. And a lot of times they see convergent evolution. So even though initially, say, 10 cells had different kind of karyotype, as they are growing, several of them, say, gained 1q arm amplification. So, and they also compared that with an actual kidney cancer tissues to see that it's the same kind of aneuploidy that these cancers have. And now these new cells that they were stressed and they were just cycling for some time, they gained the exact same chromosomal aberrations. So this is direct evidence to show that these chromosomal aberrations are actually driving survival of these cells. So now what I would want to do is have cells which have normal EGFR, mutant EGFR, do the same thing, and then let them grow and then see if EGFR mutant cells, they have a specific kind of aneuploidy.
Interesting idea.
Yeah, there was another paper by Christina Curtis where they have created TP53 mutations of different types. They literally let them grow for fucking 2 years. Then after that, those 2 years, they see very similar sequential event of chromosomal abnormality. So it's like a very sequential manner in which this evolution is happening in the TP53 background.
It's not like random.
Yeah.
Okay. Anything to wind down or?
Let's see. Anything you're excited for?
Tomorrow's plan. Excited for the barbecue.
Yes. Yeah, what motivated you suddenly?
We had these grand plans and then we settled for a house party. So I feel like we should at least do something.
Yes, the barbecue would be nice. Sounds fun. Cool.
Cool.